Welcome to this crash course on General Biology 1. Let's get started. Let's begin with evolution, the concept that populations adapt to their environment over time. Charles Darwin and Alfred Wallace both explored many islands, such as the Galapagos and the islands of Malaysia, and noticed that these places had very unique species, unlike anywhere else. They concluded that these populations of different species evolved as traits were, quote, selected for by the environment through a process called natural selection. In other words, the traits that were most advantageous to survival and reproduction were passed down through the generations. Their theory goes on to say that species change over time. Species that diverge or become separate species share a common ancestor, and populations, but not individuals, often show genetic variation and can evolve through evolution. This genetic variation is often caused by mutations, or random changes in the DNA of an organism, and these can be harmful, beneficial, or neutral. Talking of DNA, it contains many genes which have different alleles, or different forms of the same genes, which can be dominant, recessive, or neither. Each population has a certain allele frequency, which is a proportion of certain alleles in the population. Individuals that are so-called diploid have two alleles, and the combination of these two alleles make up what is called the genotype of the individual. They can be homozygous dominant and have two dominant alleles, homozygous recessive and have two recessive alleles, or be heterozygous with one dominant and one recessive allele. There are also other causes of evolution, including gene flow, or migration of individuals and their genes to and from different populations, genetic drift, or changes in allele frequencies that occur purely by chance, and non-random mating, where traits which increase chances of attracting mates are chosen for. In order for a population to not evolve over time, it must satisfy the conditions of the Hardy-Weinberg Equilibrium, which are as follows: no natural selection, no gene flow, no mutations, random mating, and a large population size. You can remember these using your 5 fingers. Your thumb to signify natural selection as you give a thumbs up and select for traits that are beneficial, your pointer finger to signify gene flow as you point individuals in the direction of new populations, your middle finger to signify M as in no mutations, your ring finger to signify marriage and non-random mating, and your pinky to demonstrate a large population size, the opposite of a tiny population, like your tiny pinky. Under these conditions, the frequency of homozygous dominant individuals can be called p squared, the frequency of homozygous recessive individuals can be called q squared, and the frequency of heterozygous individuals can be called 2pq, where p squared plus q squared plus 2pq equals 1. Using statistics and concepts called the null hypothesis and alternative hypothesis, we can test if a population is in Hardy-Weinberg equilibrium. And I would highly recommend checking out our crash course on prop stats for a more in-depth look. Populations can also undergo selection in many different ways. Stabilizing selection favors average individuals. Directional selection favors individuals that differ from the average in one direction, such as above average. And disruptive selection favors individuals that are either above or below average. Species can be identified using the morphological species concept, where members of a species can be identified by how similar they look. This can be flawed though, since individuals within a species can sometimes look quite different. So, a more robust concept is the biological species concept, where different species are species that cannot interbreed due to genetic differences. However, speciation is not always complete and can sometimes be a gray area. For example, Neanderthals and modern-day humans are typically considered separate species, but they interbred at some point in history. Now, let's change courses and dive into living organisms, into cells. Cells contain mostly water, but also ions and small molecules, and bigger molecules called macromolecules. These macromolecules are called polymers, or molecular structures composed of monomers, or single subunits. These include proteins, lipids, carbohydrates, and nucleic acids. Starting with proteins, proteins are extremely important in living organisms as components of receptors, in helping with transport, as defense molecules, as structural molecules, and as enzymes, in other words, speeding up reactions. They are made up of amino acids, where amino acids share a structure of a central or alpha carbon atom, an alpha amino group, an alpha carboxyl group, a hydrogen atom, and a varying side chain called an R group. Peptide bonds, a type of covalent bond, link amino acids, where the carboxyl group of one amino acid loses an oxygen and the amino group loses two hydrogens. The amino acids bond, and water is a byproduct produced by the excess oxygen and two hydrogens. Chains of amino acids, also known as proteins, extend from their N-terminus, or amino group at the start, to their C-terminus, or carboxyl group at the end. Amino acid side chains with oxygen or hydroxide groups in them are often polar and will stick around other polar molecules such as water, whereas those with no oxygen tend to be nonpolar. A protein is only biologically active once it folds into its so-called native structure, where amino acid side chains with nonpolar side chains tend to be buried inside to avoid interaction with the often polar environment, in other words, water. The sequence of amino acids themselves is called the primary structure, and the folding of the primary sequences within themselves into alpha helices and beta sheets is called the secondary structure. The interactions and further folding of the alpha helices and beta sheets and overall folded structure of this now so-called polypeptide is called the tertiary structure. And finally, the quaternary structure is the structure formed by the interaction and folding of several polypeptides, in other words, the final protein. The protein and its subunits can become denatured or unfolded and inactive if they're heated or experience chemical or pH changes. Next are lipids, which are a large group of molecules composed of hydrocarbons or hydrogen and carbon atoms, and which are very nonpolar and insoluble in water. They store energy, form cell membranes, and serve as thermal insulation in animals. A big subgroup of lipids is triglycerides, which include fats, which are solid at room temperature, and oils, which are liquid at room temperature. Triglycerides consist of one glycerol molecule and three fatty acids, which have a polar or hydrophilic head and a nonpolar or hydrophobic tail. They can either be saturated, where all carbons have a hydrogen and there's no kinking or bending of the chains, or unsaturated, where not all carbons have a hydrogen and there is kinking of the chains. Within unsaturated fats are trans fats, which are linked with coronary heart disease, and cis unsaturated fats, which are healthful and found in plant oils, walnuts, and fish. Another subclass of lipids are phospholipids, which consist of one glycerol molecule, two fatty acids, and a phosphate group. In an aqueous environment such as in water, they naturally and spontaneously assemble to form the phospholipid bilayer, forming the membranes of cells, where the nonpolar hydrophobic tails pack together inside and the polar hydrophilic heads face outward and interact with water. Now onto carbohydrates, which are important as the main source of stored energy within cells, structural molecules that give organisms like plants their shape, and recognition molecules inside and on the surface of cells, like sugars on blood cells in humans. Their monomers are called monosaccharides, which form the sugars themselves, or polysaccharides, via glycosidic bonds. An example are sugars such as glucose. Glucose is a very commonly mentioned carbohydrate and is a hexose with 6 carbon atoms forming a ring. There is also cellulose, an unbranched polymer of glucose, which is an important structural material in plants, starches, which are branched polymers of glucose that humans consume, and glycogen, a highly branched polymer of glucose which acts as a major energy storage molecule in animals. Finally, onto our last polymer, nucleic acids. There are 2 kinds of nucleic acid polymers, DNA, or deoxyribonucleic acid, and RNA, or ribonucleic acid. They are made up of monomers called nucleotides, which are adenine A, guanine G, cytosine C, and thymine T, where RNA has a very similar uracil U in place of thymine. The purines, adenine and guanine, consist of 2 cyclic rings, while the pyridamines, cytosine, thymine, and uracil, have 1 cyclic ring. Additionally, adenine and thymine and uracil form 2 hydrogen bonds with each other, while cytosine and guanine form 3 hydrogen bonds with each other. DNA and RNA both have 1 5-carbon sugar, 1 nitrogen-containing base, and 3 phosphate groups. They start from their so-called 5' phosphate group and are extended from their sugar. The main difference is that RNA has ribose as its sugar, whereas DNA has deoxyribose as its sugar. RNA also tends to be single-stranded and folds in on itself into unique 3D structures, while DNA tends to be a double-stranded antiparallel helix. Going back to lipids and proteins, these make up the membranes, or outside coverings, of cells. Proteins can be either on the outer portion of the membrane, or embedded inside or across the membrane. Some molecules can passively diffuse across the membrane by themselves. For example, water can flow down its concentration gradient, in other words from areas of higher concentrations of solute or dissolved substances, to areas of lower concentrations in a process called osmosis. Molecules that cannot diffuse across the membrane by themselves use proteins to go down their concentration gradient and get across using facilitated diffusion. There is also active transport, where proteins pump substances like ions across the membrane. This requires energy since the molecules are going against their concentration gradient. Many macromolecules that are too large for these proteins will cross the membrane via endocytosis and exocytosis, where they are engulfed and released, respectively, by structures called vesicles. In general, membrane proteins in vesicles allow substances to move across biological membranes and regulate the concentrations of substances inside and outside the cell, which is critical to the cell's proper functioning. Additionally, these membranes are actually fluid, constantly moving and shifting and changing. Lipids, like the steroid cholesterol, decrease this membrane fluidity, along with cooler temperatures and tighter packing of the fatty acids of lipids. Diving further into cells and their structure, we know that cells are the fundamental units of life, that all organisms are composed of cells, and that new cells come from pre-existing ones. Viruses are a gray area since they don't consist of cells, but some consider them alive. Cells either have a nucleus containing most of their DNA and are called eukaryotes, or don't have a nucleus and are called prokaryotes. All cells have plasma membranes that allow them to separate their internal environment from their external environment. Eukaryotic cells tend to be bigger at around 10 to 100 micrometers. Prokaryotic cells tend to be smaller at about 1 to 10 micrometers, and viruses are even smaller at 0.05 micrometers in diameter. Inside eukaryotic cells is the cytoplasm, which contains organelles, which are distinct parts of the cell that are each surrounded by a membrane and that serve specific functions. The endoplasmic reticulum transports materials and processes lipids. The mitochondria makes energy. Ribosomes make proteins. The Golgi body packages and ships material out of the cell, and lysosomes break down proteins. 90% of the dry weight of all biological molecules on Earth are composed of 6 atoms, nicknamed sponge, for sulfur, phosphorus, oxygen, nitrogen, carbon, and hydrogen. When molecules react to create products, these products can also react in the opposite direction and reform those products. These are called reversible reactions, and they're quite common. When there is a zero net change in the quantity of reactants and products, the mixture has stabilized in what is called an equilibrium. Cells harvest energy through a series of chemical reactions. The overall strategy is to first break down materials, or polymers, into parts, or monomers. Then, the monomers are converted to carbon dioxide and water, releasing energy. Some of the energy is released as heat, and the rest is stored in molecules called intermediates. Finally, the stored energy is used to make ATP, which can be used by cells as a direct energy source to power cells' chemical reactions. For example, eukaryotes use aerobic respiration to produce energy by converting glucose and oxygen into ATP, with water and carbon dioxide as the byproducts. Aerobic respiration starts with glycolysis, where glucose is converted to pyruvate, which is then converted to acetyl-CoA. The citric acid or Krebs cycle converts acetyl-CoA into various compounds and produces the energy intermediates NADH and FADH2 along the way. Finally, the energy stored in NAD and FAD is released in oxidative phosphorylation, where a process called the electron transport chain produces the vast majority of ATP in aerobic respiration. In the absence of oxygen, cells can produce limited energy in a process called anaerobic respiration, more specifically called fermentation. Cells must divide in order to replicate and pass on their genetic material. Prokaryotes divide using binary fission, while eukaryotes divide using mitosis and meiosis. In the eukaryotic cell cycle, cells grow and duplicate their DNA, then undergo mitosis, where one cell turns into two cells with the exact same DNA content as the original cell. This process is strictly regulated by various checkpoints along the cycle and proteins called CDK and cyclin. Mitosis occurs in all the cells of the body except for those that reproduce sexually in a process called meiosis. Meiosis produces specialized cells called gametes, or the sperm and egg. Meiosis is a two-step process that produces 4 haploid cells with half the DNA content of the original diploid cell. There are also 2 phenomena called crossing over and independent assortment, which contribute to genetic diversity and means that every individual haploid cell produced is genetically unique. The sperm and egg then fuse and produce a single cell called a zygote, which develops into an organism such as a human. In the 19th century, a monk named Gregor Mendel studied inheritance in garden peas, which were easy to grow and produce lots of female and male offspring. He crossed or bred peas that were often true breeding, in other words, homozygous for a certain trait, and always passing down that trait to their offspring. The original parental generation is called P, their offspring is called F1, and the offspring of F1 is called the F2 generation. Mendel noted that traits could skip a generation, since the peas that appeared to have the dominant phenotype could still carry the recessive allele in their genotype and pass it down to the next generation. To visualize different crosses, we use Punnett squares with the male's alleles on one edge and the female's alleles on the other edge, and the resulting allele combinations, or genotypes, inside the boxes. Here you can see an example with brown and blue eye alleles. Note that the best way to learn Punnett squares is to practice using them in word problems. There is also incomplete dominance, which is where heterozygotes have an intermediate phenotype that's a mix of the 2 phenotypes, and codominance, where both phenotypes are present at the same time. An example of codominance is blood type, where the blood type AB has surface sugars from both blood type A and blood type B. We can also use family trees, or pedigrees, to show patterns of inheritance over many generations and predict future phenotypes. Some diseases are transmitted by dominant alleles, and others are transmitted by recessive alleles. Additionally, inheritance can be sex-linked, or passed down through sex chromosomes, or autosomal, or passed down through non-sex chromosomes. Diseases passed down the sex chromosome, called the X chromosome, are X-linked and tend to be more common in males, since they will express their only X chromosome, while females have 2 X chromosomes and are often carriers. An example of an X-linked trait is red-green colorblindness. We can look at similar trees called phylogenetic trees to look at how organisms are related to each other. These start with a common ancestor and then branch off as new species are formed. When we classify species into groups, we can say that they are monophyletic, paraphyletic, or polyphyletic. Monophyletic means that they are in a group that contains an ancestor and all descendants of that ancestor. Paraphyletic means that they're in a group that does not include all descendants of a common ancestor. And polyphyletic means that they're in a group that does not include a common ancestor. Traits of different species can be classified as homologous traits, synapomorphies, evolutionary reversals, and convergent evolution. Homologous traits are traits that were inherited from a common ancestor and shared by 2 or more lineages. Synapomorphies are characteristic and unique traits shared only by the most recent common ancestor, MRCA, and all its descendants. Evolutionary reversals are when traits are lost or revert back to an earlier form, and convergent evolution is when similar traits appear independently in different species. An example of convergent evolution is the wings of bats and birds, which are very similar even though they developed independently of each other. Finally, let's talk about the central dogma of biology, which is DNA to RNA to proteins. DNA is a double-stranded antiparallel helix consisting of nucleotides. When DNA is replicated, each strand is separated and used as a template for a new complementary DNA strand. A primer is placed at the start of replication, called the 5' end, and nucleotides are added along the strand in the 3' direction. These nucleotides are the complementary pairs of the template strand nucleotides, where A's are put across from T's and C's are put across from G's. Enzymes called primase, helicase, and ligase also help in the process. In the end, 2 DNA strands are created from 1. It's also good to know that in humans, our DNA is contained in 23 pairs of chromosomes in each of our cells, of which 22 pairs are called autosomes, while 1 pair is called our X and Y sex chromosomes. Normally, females have 2 pairs of X chromosomes, and males have an X and Y chromosome. A fascinating concept is that of telomeres, which are short, repeating, non-coding sequences of DNA at the end of eukaryotic cells. Since cells shorten each time they divide, telomeres protect chromosomes by taking the brunt of the shortening when cells divide. Telomeres shorten over time as we age, and a theory is that we can slow aging by increasing the lengths of our telomeres. Moving on to RNA. RNA is created from DNA through a process called transcription. There are 3 main types of RNA: messenger RNAs, mRNAs, which are templates for protein production during translation; ribosomal RNAs, rRNAs, which help with the structure and function of ribosomes; and transfer RNAs, tRNAs, which donate amino acids to make proteins during translation. RNA is very similar to DNA, except that it is typically single-stranded, and it has U for uracil in place of T for thymine. Uracil is still the complementary nucleotide to A, though. Similar to DNA replication, RNA polymerase adds on complementary RNA nucleotides across from a DNA strand, creating a complementary mRNA strand. This strand is then modified in a 3-part process. First is 5' capping, where chemically modified guanine is added as a cap to the 5' end of the mRNA sequence. Next is polyadenylation, where 100 to 300 adenines are added to the 3' end. Finally, we have RNA splicing, where certain parts of the sequence called introns are spliced or cut out, and the remaining parts called exons are left behind. This processed, mature mRNA sequence is then converted into proteins in a process called translation. tRNA reads the mRNA sequence and then produces the corresponding amino acids. Each amino acid is coded by a unique group of 3 nucleotides, and strung together, these amino acids compose peptide chains, or proteins, which play a critical role in the structure and function of cells. Going back to mRNA, mRNA has become highly relevant in recent months due to the creation of mRNA vaccines in the fight to end the COVID-19 pandemic. These vaccines inject the body with mRNA that codes for the spike proteins of the SARS-CoV-2 virus that caused COVID-19, allowing the body to recognize it and build immunity to protect from future infections. And who knows, maybe mRNA has many more potential uses that we have yet to explore. And that's it. I hope you found this crash course on General Biology 1 helpful.